In this video, we will discuss about reactive oxygen species and its effect within the cells. So let us start with a general uh, central dogma of the cells. That is, DNA is present in the chromatin that is considered as a genetic material. And what happens is first transcription occurs where DNA is converted into the RNA, and then after that RNA is converted into the functional protein with the help of translation. And this functional protein helps in the metabolism within the cells. That is the food or whatever the carbohydrates, protein, fats, xenobiotics, tobacco, ethanol, whatever we consume is converted into the CO2 and waste with the help of oxygen. And during this conversion, there is some byproduct are also formed while oxygen is reacting to all this all these things, and these byproducts are called collectively called as ROS, that is reactive oxygen species. And in the normal human being, this reaction is always in an equilibrium state in such a way that the amount of ROS is very low or within the acceptable limit for a healthy individual. So after that, this reactive species then are being controlled in the normal or healthy cells, and there are other things, other functional proteins are also produced that helps in controlling this reactive species are antioxidants, nutritional cofactor enzymes. And apart from this byproduct, there is also this factors, tissue growth factors or other TGF alpha growth factors will helps the cells to differentiate into a particular form. And what is this cell cycle differentiation or adaptive response means? We will understand this from the another figure on the right side in brief. That is, first it starts from very initial state when the sperm or fertilize with the interact with the egg and fertilization occurs, and then zygote formation occurs, and finally it leads to the formation of ICM, that is inner cell mass. And this inner cell mass it contains some blastocytes, and these blastocytes will finally converted into the embryonic stem cells, and these embryonic stem cells with time produce some growth factors that is shown by this arrow on the right and left hand side figure. And in the presence of certain growth factors, and based on the growth factors and the quantity and the environment in which the cells are present, it leads to a particular form of differentiation. And this differentiation leads to the formation of final committed cell lines, and these differentiation conditions are different to form a different form of cell lines. For example, if bone marrow tissues are formed, these differentiation conditions are different than the kidney tissues or kidney cells. This is called differentiation, and ICM can also be directly differentiated to form this final committed cell or adult stem cells. Oh, embryonic stem cells first form adult stem cells, and after that they will convert it into uh, progenitor cells, and then into the somatic cells. And after that they will form into the committed cells. And from these committed cells, scientists have identified that they can be reprogrammed into the induced pluripotent stem cells, and these are equivalent to the embryonic stem cells. And after that, this can be further differentiate into the specific environment or the growth factors to convert them into a final desired form of cells. So this strategy can be used to treat the patients that have uh, different that have unhealthy cells in a particular part of body. So that cells can be replaced with newly differentiated cells from induced pluripotent stem cells. So this is the basic idea about how the stem cells are functions, and how we can use this strategy to cure the person. So and this whole thing is this natural process, that is ICMs converted into embryonic stem cells and then into the differentiation is represented by this line. Now let us come to the topic, that is what will happen if in a condition where ROS is uh, uncontrollable or it is quite high. It will lead to various problems, and this is maintained here, that is DNA damage, mutagenesis, carcinogenesis. Senescence means it is a state where cells has a metabolic activity, metabolically active, or and but is has stopped its growth, and all this will finally result in DNA damage, and this will activate the DNA repair mechanism that can be activated by P53 signal. So, so that is the reason the reactive species are needs to be controlled, and if these reactive species are increased beyond the limit, it will cause various problems, and that might some of them might be harmful or fatal to the human beings. And in this figure, it has shown one of the example that how this reactive oxygen species uh, cause a problem. We will not go in deep into this signal, what is the purpose of this signal. We will just understand what are the problems it will cause. So if the ROS, that is reactive oxygen species, is beyond a certain limit, it will prevent these signals that is essential for normal function, that is glycolysis, BMI1, HIF1, and SOD catalase. These things will be stopped, and what it will promote? It will promote DSB, that is a double stranded break, DNA double stranded break. Uh, that is, it will lead to the breaking of DNA, DNA's double strand, and this will activate P53. P53 will take the decisions whether it needs whether the DNA break is uh, within repairable limit or not. If it is not in the repairable limit, it will uh, direct the cells towards apoptosis, that is program cell death. And if it is within reproducible limit, what it will does, it will arrest the cell cycle till the DNA breakage is repaired, so that this thing will not spread across other cells. And at the same time, this ROS will lead to different form of differentiation, as it will change the environment in which the cell is present, and it will also cause senescence state through P50, P38, P16, and senescence, as we have discussed, as we have known from just a few minutes ago, that senescence is a state where cell growth rate is arrested, however it is metabolically active. And this is the timeline that it shows how the increase in oxygen or oxygen IR and stress in combined will cause problems within the body. The increase in oxygen IR and stress will cause more ROS within the body, and this will reduce the glycolysis formation with the glycolysis reaction to uh, OXPHOS, and as it is shown here, it will block several signals, that 1, 2, and 3. There's BMI1, HIF1, and this glycolysis will also stopped. And this thing results in more ROS formation. Uh, these things are the result of more ROS formation, that is with the increase in oxygen amount will leads to more reactive oxygen species formation within the cells. And finally, what it will affect this, it will affect the homeopathic stem cell self renewal capacity. As we know from previous stem cell videos, that stem cells has a capacity to self renew themselves. However, that thing will be affected because of this increase in ROS uh, within the cells, and it will also affect, it will cause further activation of signals that are not uh, that are not advantageous to the cells or the patients who are experiencing this. That is, it will uh, affect this. It will increase this apoptosis cell death. It will cause increase change the state of the cells to senescence. It will increase its differentiation to undesired committed cells. And at the same time, this self-renewal proliferation. It will promote self-renewal proliferation. It will promote self-renewal proliferation. So what it means is that uh, HSC cells that are not in a uh, healthy state will self-renew itself, and uh, this might lead to further spread of this unhealthy cells across the body, or we can say that metastasis. And here the cells will remain quiet. And one thing that I have to say, or I have to correct, that all these state are relative to the condition at which it is present. For example, at this state where oxygen stress is low, cells are at quiescent state, that is almost at healthy state. And in this state now, cell proliferation is within limit, or self renewal proliferation are of homeopathic stem cells are within limit. And after that, as the oxygen IRL stress increases, it will cause problems. That is, it will differentiate unhealthy cells into undesired committed cells, and finally it will change the state of the cells to senescent state. And then the further increase in the oxygen IR and stress will cause the cells to proceed towards apoptosis cell death. So that's all for this video. Thank you for your time.